This presentation will cover Chapter 8, Environmental Sample Collection and Processing. In terms of different environments, this presentation will focus on soil, water, air, and fomites. Soil is a complex system. The overall sampling strategy will depend on many factors, including the goal of analyses, the resources available, and the site characteristics. Because there are so many different factors, it's important to begin with a strategy that will ensure quality assurance is addressed. This can be done by creating a quality assurance project plan according to the guidelines. The guidelines are as follows. Sampling strategies, meaning decide on the number and types of samples, locations, depths, times, and intervals. Sampling methods, what type of specific techniques and equipment are to be used. Sampling storage, meaning what types of containers, preservation methods, and maximum holding times do you wish to use. And sampling techniques. A very important factor to consider when creating a project plan is what level of soil you plan on sampling. Surface soils will have different methods than subsurface soils, and it's important to know the difference in sampling techniques between the two. Collection of surface soils may often be done using a shovel or a soil auger. An auger is more precise because they ensure that samples are taken to the exact same depth on each occasion. This is important as several soil factors can vary considering with depth, such as oxygen, moisture content, organic carbon content, and soil temperature. If collecting for microbial analysis, the auger can contaminate the sample. So either the outer layer of the sample is scraped off and discarded before analysis, or the auger is cleaned in between each collection. The rhizosphere is of special interest to soil microbiologists and plant biologists because of the enhanced microbial activity and specific plant-microbe interactions. In this case, roots are carefully excavated and shaken to remove the bulk of non-rhizosphere soil. And although this is a crude sampling mechanism, it remains intact to this day. However, as a result, the collection remains as an experimental limitation. Composite samples can be attained by collecting equal amounts of soil from samples taken over a wide area and placing them in a bucket or plastic bag. The whole soil mass is then mixed and becomes a composite sample. In some instances, a series of experimental plots of fields need to be sampled to test the effect of the soil amendment. In this case, they can be further divided down into plots. In random sampling, you choose random points within the plot of interest, which are then sampled to a defined depth. Transect sampling involves collection of samples in a single direction. Two-stage sampling is when an area is broken into regular subunits called primary units, and then within each primary unit, subsamples can be taken randomly or systematically. Two-dimensional sampling does not give any information about changes in microbial communities with depth. Therefore, three-dimensional sampling is used when information concerning depth is required. Drill rigs are necessary for sampling subsurface environments. This increases the cost of sampling, especially for the deep subsurface soils. For a saturated soil system, an air rotary drill is used. A large compressor is used to force the air down a drill pipe, out the drill bit, and up outside the borehole. As the core barrel cuts downward, the air serves to blow the borehole cuttings out of the hole and also to cool the core barrel. This is important because if the barrel overheats, microbes within the sample may effectively be sterilized, posing difficulty for subsequent microbial analysis. To maintain sterile conditions and prevent contamination from surface air, all air used in the coring process is pre-filtered through an HEPA filter. For an unsaturated soil system, the borehole must be held open, so a hollow stem auger drill with a push tube is typically used. For the processing portion of soil collection, all samples should be stored on ice until processed and analyzed. They should also be sieved to remove large stones and debris before analysis. A typical process that is used is called soil washing, which is where the soil is saturated with sterile water until only heavy particles remain, and then the sample is observed under a microscope. Analysis generally involves cultural assays and plating methodology, or community DNA analysis, which is a process that aims to observe the influence of the amendments to the microbial communities. Water is often easier to collect than soil, but it involves harder processing. In order to process samples for viruses, the process of filtration is typically used. There are two main types of filters, which are electronegative and electropositive. If you are sampling a very large volume of water, a virus absorption or Viridel filter is used. The virus is then recovered from the filter through elution, as well as a reconcentration step to reduce the sample volume before assay. Detection of viruses are then done by cell culture or PCR. When processing water samples for bacterium, there are two processes commonly used. Membrane filtration, which involves the collection of concentration of bacteria via filtration, and most probable number, where the samples are not processed prior to being analyzed. Another important process in water sampling is processing for protozoan parasites. This involves large volumes of tap water or surface water passing through a filter. Filters can be pleated cartridge or made of foam. If it is a pleated cartridge filter, an elution buffer is added to the filter, placed on a shaker, and agitated for 5 minutes to release the cyst. This is then followed by centrifugation and a resuspension in a buffer. If a foam filter is used, a phosphate buffer is added and the protozoa are squeezed from the flexible filter with a plunger. Choosing a device for air sampling collection is dependent on the type of collection method you wish to use. Impingement involves trapping of airborne particles in a liquid matrix. Impaction involves the forced deposition of airborne particles on a solid surface. Filtration is the process of trapping of airborne particles by size exclusion. And deposition involves the collection of airborne particles using only naturally occurring deposition forces. The most common devices used for air sampling collections are the AGI 30 impinger, the SKC impinger, and the Anderson 6-stage impaction sampler. Fomite collection generally uses two different methods. The first is the use of Rodock agar plates. These are petri dishes in which the agar fills the entire dish to produce a convex surface, which is then pressed against the surface to be sampled. The second is the swab and rinse method, in which a sterile cotton swab is moistened with a buffer and rubbed over an area to be sampled. The samples are then either incubated or washed, and the colonies are counted or PCR is performed.